All right, thanks very much for having me here. I'm always delighted to talk to the NREC audience, and I'm glad you're all energized with the cookies too to get us through this last talk. I know I'm the only thing standing between you and the poster session, so we'll be efficient here. Okay, so I'm going to talk about our progress in our projects to breed crops for microbiome-associated phenotypes that contribute to nutrient retention. And I saw this on John and Andrew's talks, and so I thought it was a great idea to put in some acknowledgments up at front too. So I'm here representing a really terrific interdisciplinary team that covers areas like environmental microbiology, nutrient cycling, crop physiology, and genetics. And we are supported by funding for both the basic and applied research aspects of this project. And I'm particularly appreciative of the sustained NREC funding that's allowed us to explore the microbiome-associated phenotypes in maize that can contribute to nitrogen retention and sustainability. And so we've leveraged this NREC investment over our maize program over the last 5 years to secure over $6 million in federal funding from ARPA-E and USDA, with $1.5 million more in pending proposals. And so this is a return on investment so far of 5.4 to 1. Okay, so like Andrew, I want to start off with the nitrogen cycle too. So as you know, modern agriculture depends on nitrogen fertilizers to feed a growing population and fuel bioenergy production. But these nitrogen fertilizers come at a cost. We produce nitrogen fertilizers using the Haber-Bosch process, which converts atmospheric nitrogen to plant-available ammonium. But breaking that nitrogen double bond or triple bond is incredibly energy intensive. And this process accounts for something like 2% of global energy use. And then even after applying the nitrogen fertilizers, we lose a significant portion through microbial processes like nitrification and denitrification. So what's happening to all that nitrogen that we're adding to the environment? So ammonium is retained in the soil, and that's available for plant use, but microbes can also use it for an energy source. And the process of nitrification transforms that ammonium, which would stay put in the soil, to nitrate, which is readily leached from the soil and transported to aquatic ecosystems. And then through the process of nitrate can also be denitrified and can be lost to the environment as gaseous products like nitrous oxide. Okay, so since microbes are responsible for all of the transformations in the nitrogen cycle, we are looking at managing the activities of soil microbes to assist with nutrient retention to improve agricultural sustainability and also our water quality. So we're looking at factors that control soil microbial communities as environmental microbiologists and microbial ecologists. And so, so we want to identify the drivers that control microbial community composition and activity. And then of those factors, what are some that we can actually manage or manipulate? And so we see things like agronomic management practices or crop breeding as important levers for this process. And so eventually we want to get to the point where we can predict the impact of management interventions on the soil microbiome. Okay, so what is it that we want to manage then? What do I mean when I'm talking about sustainability? So ideally, we'd like the microbes to assist with nutrient provisioning, so things like nitrogen fixation and nitrogen mineralization. But beyond that, we'd also just like the nitrogen that we add to the field to remain there. So we're interested in microbial functions that impact nutrient retention, things like denitrification, nitrification, and DNRA. Dissimilatory nitrate reduction to ammonium. So we'd like to reduce the amount of denitrification, we'd like to reduce nitrification, and possibly even increase dissimilatory nitrate reduction of ammonium, which would take nitrogen from this mobile form of nitrate to the less mobile form of ammonium. Okay, so plants recruit and assemble their microbiomes by filtering the microbial species from the soil around them, and that soil microbiome is influenced by environmental drivers, edaphic factors like soil texture, pH, moisture, redox, and soil types. And these edaphic factors vary a lot across a lot of different regions and soil types. But the recruitment of the soil microbiome then is also or the recruitment of the rhizosphere microbiome is also controlled by plant factors. So like the root system architecture alters the surface area and the volume of the rhizosphere, controls the flux of carbon into the root environment. And plants can release metabolites to recruit a wide diversity of microbiota to the root zone. So these are all factors that shape the assemblage of microbes that might colonize the plant's root surface or the interior of the plant. And importantly, plant genotype plays a role in the action of these ecological filters. It determines the root characteristics, the metabolite characteristics. So plant genotype controls that, so it suggests that it might be possible to intentionally modulate the ecological drivers of the soil microbial community, modulate the community composition and the activity of the rhizosphere microbial community through plant breeding. So basically breeding plants for microbial phenotypes. So to start to study this, we surveyed the microbial communities associated with a variety of corn genotypes. Okay, and before I get to that, just want to give a quick overview here. So we're interested in the microbiome because it can help the plants to acquire nutrients. Also, the microbes are pretty famous for causing plant disease or controlling plant diseases too. So it impacts plant health, and in natural systems it can play a role in plant competitive interactions too. But in modern agriculture, we have replaced a lot of these microbiome functions through nutrient input and agronomic practices. So we hypothesized that the process of selecting for high crop yields in environments with increasingly high levels of nutrient inputs might have inadvertently led to large changes in root traits and the associated rhizosphere microbiome, such that the microbiome-associated mechanisms for nutrient acquisition and nutrient use efficiency might have been compromised. So the plants, we hypothesized that the plants became less dependent on plant microbe interactions as we bred them for current high input environments. And in fact, this might actually even limit the ability of modern crops to take full advantage of some sustainable management approaches that we might be interested in developing. So we started off by asking what has domestication and crop selection meant for the microbiome and its functions? So during the Green Revolution, maize selection and breeding took place under increasingly higher nitrogen fertilizer levels. There we go, you can see that. And so when released from the need to partner with soil microbes for nutrient acquisition, we wondered what happens to the microbiome and its function. So here the selection has taken place under conditions where we have reduced reliance on any microbes for nutrient acquisition, and so there's no selective pressure to maintain the microbiome functions related to nutrient acquisition. And so what does that mean for the microbiome? And so one really great thing about maize is that we have access to germplasm spanning the history of domestication and breeding. And so we took advantage of that to compare the microbiome over a germplasm chronosequence that spanned the Green Revolution. And so that's what I'm representing here on the x-axis here. Each of these blue lines represents the year of germplasm release of a different maize line. You can see that they were developed under different levels of nitrogen fertilizer. Okay, so this was so this first study was a greenhouse study where genotypes representing germplasm development over this time were grown in maize-naive soil. And because we didn't want to study the microbiome of nutrient-starved plants, we did fertilize these all at the same level. And so for our first result, we saw that the maize rhizosphere differed across these genotypes that were developed in different years. And then we wondered, what does that mean for microbial functions? So we used, so we identified the microbial communities that, the specific microbial populations that were changing across this germplasm chronosequence, increasing or decreasing in relative abundance over or among these different genotypes. And then based on those microbial taxa that were changing over time, where our timeline on the x-axis corresponds to the year of release for those maize genotypes developed for higher nitrogen fertilization, we looked at the predicted functions of the microbial community. And so of interest for sustainability is this decrease in genes that contribute to nitrogen fixation or to nitrogen mining of nitrogen through mineralization. And so this agrees with our hypothesis. So we're seeing a decrease in these potentially sustainable functions over this period of germplasm development. And then we also showed that the predicted functions related to unsustainable nitrogen cycling functions, so things like denitrification and nitrification, those were increasing in the more modern lines too. So again, recall that these were identical growth conditions, the same starting microbiome for each of these different maize lines. And so it was particularly interesting, we saw the decline in the recruitment of nitrogen-fixing bacteria on the previous slide. In our more recent germplasm. So we were fertilizing these plants. They were definitely not benefiting from nitrogen fixation, but we still saw differences in colonization of these potentially beneficial microbes in the rhizosphere. And similarly, on this slide, it's not the case that the more recently developed lines were getting more nitrogen fertilizer. That might explain why we'd seen enrichment in these functional groups. Instead, this effect was driven by the plant and its root characteristics and how it recruits the microbiome. Okay, so to summarize just some of our early efforts, the maize rhizosphere microbiome reflects this germplasm chronosequence. We saw nitrogen cycling genes in the rhizosphere altered across this germplasm chronosequence, likely driven by increased fertilization rates and selection acting concurrently over time. Okay, so those previous results were just some changes in the microbiome recruitment over past several decades when maize had already experienced a good deal of genomic changes through domestication and breeding. But what if we went even further back in time? What sustainability traits got left on the editing room floor during domestication and the change from our teosinte here to modern maize? And what can we rediscover by examining the ancestor of modern maize? And so this change from teosinte to modern maize represents a change in the root structure, the seed product, the entire architecture of this plant. And what I'm showing here is relevant not just for food crops, but also for other bioenergy crops too, where we're just at the beginning of purpose breeding, and we can make decisions for maintaining or optimizing desirable microbial functions that contribute to sustainability. So maize here could be, in this instance, sort of a case study where we can look at the outcomes of long-term selection for above-ground traits on the microbiome functions related to sustainability. Okay, so for our longer-term perspective, we compared 6 different genotypes of teosinte with 6 modern maize lines, all grown in the same maize-naive soil. So each of these points represents a microbial community collected from bulk soil or the rhizosphere of a plant. The closer they're plotted together, the more similar the microbial communities are. The further apart they are, the more different the microbial communities are. Okay, so from this starting microbial inoculum, you can see in brown here, the ancestral and the modern genotypes selected different assemblages of microbes. So they started with all the same. They recruited different microbes. So the first thing we asked is, okay, well, what does that mean for function? So then we took this to the field. We followed up with a field experiment where we demonstrated that nitrification was significantly reduced in our teosinte lineages. Okay, so that's great, but nobody wants to grow teosinte, but it's an intriguing phenotype. Okay, so this phenotype of suppressing nitrification has actually been identified in other grass species, particularly sorghum, but also landrace, wild, and weedy grass species. And it's a trait that's mediated by the plant roots. Grasses are releasing exudates into the root zone, and this chemically inhibits the enzymes within the nitrification pathway. So very similar to what Andrew was just talking about with nitrification inhibitors. But in this case, it's the plant roots that are producing the nitrification inhibitors. So the plants are producing an exudate here, and then that is inhibiting the enzymes that transform ammonium to nitrate. Okay, so this seems like a great trait to recover in modern corn. And happily, another advance of the Green Revolution was the development of genomic and molecular tools that allow us to target plant microbiome assembly mechanisms and allow for precision breeding for microbiome-associated phenotypes. So we took advantage of an existing maize teosinte panel of near-isogenic lines. So to narrow down the portion of the teosinte genome that might be responsible for the suppression of these undesirable nitrogen transformations. So in this panel of near-isogenic lines, maize and teosinte were crossed, and then back-crossed repeatedly to B73 to end up with a panel of near-isogenic lines that each have small portions of the teosinte genome in a modern inbred B73 background. And so we used a panel of these that spans the teosinte genome in order to find the portions of the teosinte genome that confer this trait of interest. So, and we used a field experiment to screen that panel. And the first thing to note is that we reproduced our previous result showing suppression of nitrification in teosinte. Most of our near-isogenic lines also exhibited the modern maize phenotype, which makes sense as they are largely B73. But we were successful in identifying 2 near-isogenic lines that exhibited the teosinte phenotype of suppressed nitrification. And so that helps us narrow down our search for the mechanism for this phenotype. So we've got this trait now being expressed in a modern maize background, which is much more useful than in teosinte. Okay, and so we're continuing to try to narrow down our this genomic region. The teosinte introgressions aren't quite as tidy as I showed in my cartoon. We still need to have a lot of work to do to narrow down the genes responsible for conferring nitrification suppression, but we're working on that. We're back crossing additionally to B73 to narrow that down, and we can see that our progress here, we're retaining the nitrification suppression phenotype. We can see that nitrification is reduced by something like 40% in our recombinant lines. And this is correlated with an increase in plant nitrogen accumulation. So what that means is the plants are suppressing nitrification. That retained nitrogen is then available for plant uptake. So it keeps more of the nitrogen around for the plant. And we're working to continue to narrow down the genes and also identify the root compounds responsible for this trait. With input from the NREC Research Council, one of our objectives was to determine if these teosinte genes would interfere with yield. And that was identified as a really important objective for introducing this trait into modern maize hybrids, and one that needed to be addressed in order to implement biological nitrification inhibition as a strategy to reduce nitrogen losses. So to do this, we generated experimental hybrids. We crossed our B73-based nils with non-stiff stock inbred lines. We generated a panel of different hybrids. I'm just showing a few of them here. But the important things to note are that we demonstrated that the hybrids retained the nitrification inhibition phenotype, and that the teosinte introgressions did not interfere with yield. And then, similar to what we saw in our greenhouse studies, we see that out in the field too, that the inhibition of nitrification leads to improved nitrogen uptake by the nitrogen accumulation by the plants. Okay, and then we even saw we even saw a slight increase in yield due to our BNI trait in the most recent field season. And then because less nitrification means that there's less nitrate to be denitrified, we also measured a reduction of nitrous oxide production in our nitrification inhibiting hybrids as well. So we're reducing nitrification, reducing N2O production, and improving crop nitrogen uptake. Okay, so we've demonstrated that over thousands of years of domestication and breeding for yield, we lost key traits that allowed maize to partner with soil microbes to manage nitrogen efficiently. And so by reintroducing this nitrification inhibition trait from teosinte, we're developing some novel maize lines that can inhibit these nitrification in the root zone, keeping nitrogen in the soil longer. And if you want to chat with us more about nitrification inhibition, you can visit Sakshi Padal's poster later this afternoon. So switching to another part of the nitrogen cycle, I also mentioned earlier that inhibition of denitrification was another desirable phenotype that could reduce nitrogen losses and contribute to nitrogen retention. So our field experiments with the teosinte demonstrated that denitrification is suppressed in the wild teosinte genotypes. And if you've ever worked with denitrification, to have results, field results with this small of error bars was pretty amazing. So, this is important because this is another mechanism of nitrogen retention of available nitrogen and also reduced production of greenhouse gases. So this biological denitrification inhibition trait is less well characterized than the nitrification inhibition, but it has been described in a Japanese knotweed, an invasive plant, as a mechanism to compete for nitrogen. So the Fallopian species produce root exudates, these procyanidins. They are compounds that inhibit bacterial denitrification. They suppress nitrate reductase activity and reduce the abundance of denitrifiers, and allowing the plants to increase nitrogen acquisition. Now, these procyanidins are a class of polyphenol from the flavonoid family, and maize can produce these compounds. So we hypothesize that this may be the mechanism at work in teosinte for inhibition of denitrification. Okay, so our screening of the maize teosinte near isogenic lines identified several lines with the teosinte phenotype of reduced denitrification, which we're following up on with similar approaches to our BNI work. So our BDI work is a couple of generations behind the BNI work, but we had our first field trial with them in 2023. And though an early season drought in 2023 made it a challenging field season for studying the nitrogen cycle, we did show that the near isogenic lines with the denitrification inhibition traits had reduced N2O emissions at some time points, and this appears to make the nitrogen more available for plant uptake. And we repeated similar results in 2024. So reduced N2O measured at some time points, and then more nitrogen available for the plant. And we had BDI hybrids in the field in summer 2025, and we're testing the BDI hybrids at multiple nitrogen levels. At the high fertilization rates that I'm showing here, we're showing that we're losing less of that nitrogen as N2O. And we expect that the retained nitrogen should be available for the plant and improve nutrient use efficiency, perhaps yield, or at least the improved nitrogen uptake should allow us to maintain yield with less fertilizer. If you would like to chat more about BDI, visit Holly Anderson's poster, and we can talk you up about that. Okay, so combining our current work with a previous NREC-funded project, we're investigating whether inhibiting denitrification might favor dissimilatory nitrate reduction to ammonium, DNRA. We know from our prior NREC project that there are conditions under which both DNRA and denitrification are possible. But if we block denitrification, could we improve DNRA and reduce our leaching losses further? And this is a project that we have in progress this spring. So we leveraged these microbiome-associated traits to apply for a $5 million grant through the U.S. Department of Energy. And with collaborators at North Carolina State and Oak Ridge National Lab and University of Arizona, we're working on combining plant traits for these microbiome-associated traits with other plant traits like enhanced nutrient use efficiency, and then also best management practices to reduce fertilizer inputs and N2O production. The goal is to reduce N2O production by 50%. N2O loss isn't the most significant pathway of loss for nitrogen from farm fields, but our nitrification inhibition will help reduce leaching losses, and also just by allowing reduction of fertilizer, we'll achieve some of those goals as well. And then the BNI and BDI traits that I showed are just from one teosinte progenitor. So we're also planning on screening a panel of near isogenic lines generated from over 80 teosinte lines. This is actually our colleagues at NC State are doing this. So they're looking for additional variation in these traits that'll allow optimization. So fertilizer represents something like 40% of a farmer's operating costs, and yet we lose so much of it through microbial conversion to nitrate or to N2O. And so by improving nitrogen efficiency, we don't just reduce pollution, we lower energy demands and carbon emissions across the entire biofuel and crop supply chain. And so, let's see. Okay, so we call this stack of traits nSave, and our plan is to stack these traits, these teosinte traits that we lost in modern corn to inhibit nitrification and denitrification. And I gave it away too. I mentioned that we were pairing with other best management practices. So we grow a lot of corn. There's like 95 million acres of corn planted this past year in the U.S. and nearly 500 million acres globally. So this work has some pretty big implications. And even a modest reduction in nitrogen loss when amplified across the scale would have a huge impact on environmental quality, would scale pretty well. So let's see, I'm happy to take questions, but before I take questions, I just want to promote one more poster. We are also interested in phosphorus retention too. So come chat with us at Aislinn Geedy's poster if you'd like to learn about our work in phosphorus retention.